Hello everyone. Today we'll learn about transcription. So we're doing molecular biology. We're doing what is known as molecular biology. So under molecular biology, we are doing this topic called transcription. But before we understand transcription, let's try to look at what is known as the central dogma. The central dogma of molecular biology. So what is it about this central dogma of molecular biology? So when you're talking about the central dogma, of molecular biology, we are simply referring to the processes which take place for DNA to be converted into proteins. Okay? But there are certain processes which must occur. So the first thing is that DNA has to copy itself. So DNA to DNA. Then this DNA changes to RNA, has to change to RNA, has to make a copy of RNA. Basically, or specifically rather, that is, let's specify, it has to make a copy of mRNA. This mRNA then has to make what is known as proteins. Okay, so this is simply the central dogma of molecular biology. So the process by which DNA makes a copy of itself, that is known as replication. Okay, so when you're talking about replication, to replicate is just to make a copy. So DNA makes a copy of DNA of itself, that is replication. Then the process by which DNA makes a copy of mRNA or RNA rather, that is known as transcription. Then the process by which mRNA has to make proteins, that is known as translation. So we need to understand this for you to understand transcription. Now let's delve into transcription. Since we have understood what is known as central dogma of molecular biology, so what is transcription? Okay, so what is transcription? So when you're talking about transcription is just a process by which a segment of DNA has to make a copy of RNA. Okay, so a segment of RNA, of a DNA rather. Okay, a segment of DNA makes a copy of RNA. And to specify, this is mRNA. It is messenger RNA, okay, not just any other RNA. So we are saying it is just a process by which a segment of DNA, it is not the entire DNA which has to be transcribed. Okay, it is just a segment, a small portion of DNA makes a copy of RNA. So basically, that is what is known as transcription. Okay, let's look at, but why transcription? But why? Why should DNA be transcripted? Okay, why should DNA be transcripted into RNA? You see, this is because when a certain mutation occurs in DNA, it is more dangerous, okay, or it has more effect than when it occurs in RNA. Okay, so when there is a certain mutation, so when you talk about mutation is just a sudden change in the expression of a certain characteristic. Okay, a sudden change. It could be caused by a mutagen. Okay, it could be caused by a mutagen. A mutagen is simply a substance that causes mutation. Excuse me for that. Okay, so that is a mutagen, a substance that causes mutation. So there's a certain country where, due to the same mutagen, it has caused, due to some dangerous weapons, okay, dangerous weapons which affected that country, it has left many children to be born, or it has made many children to be born with some defects, okay, or rather should I say with some anomalies or disorders. Why? Because that weapon had to affect the DNA. It affected the DNA, so the DNA has been mutated, and it has led to children being born of certain anomalies, okay, certain challenges due to that same weapon. Okay, so that is how dangerous mutation can be if it occurs in DNA. It simply affects the genetic composition of an organism. Okay, now we know that for DNA to be converted to RNA, what has to be done is that this strand of DNA has to split. And from DNA, we said that there's an enzyme called helicase. The enzyme helicase is the one which is able to split or to break these bonds so that the two strands are able to separate. Okay, so that is what you need to understand. Now let's look at the steps of transcription. What are the certain steps of transcription? So the first step we'll look at is insertion. We even looked at insertion when you're dealing or when we are discussing what is known as DNA replication. We looked at insertion, and we said when you're looking at insertion is just the beginning or the starting, okay, the starting of the process. So we said for insertion in DNA replication, it is the starting of replication, but in RNA, in transcription, the changing of RNA to make a copy of the changing of DNA to make a copy of RNA, insertion is simply the starting, okay, the starting of the process of transcription. So basically that. Then the second process you must look at, it's elongation. So you must understand elongation also. When you're talking about elongation, we said the other name for elongation sometimes they can call it polymerization. because with elongation, what you're simply doing is you are making a polymer, a long chain. Okay, this is making a long chain. So no wonder it is called sometimes as polymerization. Then what do we call the third step? The third step is simply termination. Okay, we said termination is simply the ending of the process. You now end. You need to end the chain after you have polymerized it. So that is the third. The third process. Yes. Then the last process is known as post-transcriptional. Okay, post-transcriptional. Post-transcriptional. We talked about modification during replication, but this one is post-transcriptional modification. Okay, so post-transcriptional modification occurs in what is known as transcription, RNA transcription. That's where post-transcription occurs. Okay, so basically that is what you need to understand. Now let's look at where transcription occurs or happens. So we have two types of cells known as prokaryotes or prokaryotic cells, and we also have what are known as eukaryotes or eukaryotic cells. Eukaryotes or eukaryotic cells. So in prokaryotes, for prokaryotes, they don't have a true nucleus. Now where is this transcription going to occur? So it occurs in an organelle known as the cytosol. Okay, known as the cytosol or the cytoplasm rather. Let's make it simple. Okay, then for eukaryotes, since these guys have the true nucleus, so transcription occurs in the nucleus. Okay, as simple as that. Then you need to look at the enzyme which does most of the processes of transcription. Enzymes. Okay, enzyme which does most of the processes enzyme which does most of the processes of transcription. Okay, of transcription. Okay, like that. So the enzyme which does most of the processes of transcription, that is known as the RNA polymerase 2. It is RNA polymerase 2. Okay, the RNA polymerase 2 is the one which does most of the processes of transcription. Now let's look at the steps of transcription in depth. We said the first step is known as insertion, right? So how does insertion occurs during transcription? So insertion during transcription is facilitated by what is known as insertion factor. Insertion factor. So this insertion is facilitated by this what is called the insertion factor. So what does this insertion factor do? This insertion factor locates what is known as the promoter. Okay, it locates the promoter. So what is this promoter? A promoter is just a region rich in what adenine and thymine. Okay, a region, let me write that. Okay, so a promoter is just a region. It is just a region rich in A T bonds. Okay, AT bond. Why AT bonds? Because when you look at AT bond, they are made of just two hydrogen bonds. Okay, they're just two hydrogen bonds, so it is easy to separate them than in this type of bond, cytosine and guanine. This cytosine and guanine, there are three hydrogen bonds there. Okay, so this is difficult to separate, but when you're looking at a bond between adenine and thymine, it's just two hydrogen bond and it is easy for it to be separated. Okay, so a promoter, what it does is that it locates this same region. Okay, it locates this same region which is rich in adenine and thymine. Okay, so basically that. Then after a promoter is located, a primer is laid. Okay, a primer is laid. So once a primer is laid, okay, just after a primer is laid, we talked about this enzyme which is known as RNA polymerase 2. Now what does the RNA polymerase to the RNA polymerase 2, what does it do? So this RNA polymerase 2 starts the unwinding. Okay, it unwinds the DNA. And what happens now? Insertion now occurs. Okay, insertion simply begins. So where can I write this? So once this happens, then insertion simply occurs. Okay, okay, so after the RNA polymerase 2, after this RNA polymerase 2 unwinds the DNA, then insertion occurs. Okay, have you seen that it is very simple? So this is the first process. We are done with the first process. Okay, we are simply done with the first process. What did we say about the first process? The first thing is that the insertion factor has to locate what is known as the promoter. I'm trying to go through so that you see how simple this thing is. Okay, the insertion factor has to locate this region, the promoter region. Now, what is it about the promoter region? This promoter region, we've said that it is rich. It is rich in what? In adenine and thymine. It is rich in those nitrogenous base, adenine and thymine. Those are nitrogenous bases. Okay, so it is rich in those nitrogenous bases which contain two hydrogen bonds. So basically it is easy to separate those two hydrogen bonds than a region which has three hydrogen bonds. For example, the bond between cytosine and guanine. But for it to locate the promoter, a region rich in hydrogen, in two hydrogen bonds, just the adenine and thymine, it will be very easy for it to get separated. Okay, it is simply very easy. Now let's look at something interesting here. Okay, let's look at something interesting. So let's look at the second now process. The second process is, we said it is elongation. And elongation, we said the other name is polymerization. So elongation is simply polymerization. Let's look at this strand. Let's look at this strand. Okay, let's say we have a 3' there, we have a 5' there, we have, since these strands are bidirectional, 5' there, 3' bidirectional, I simply mean that these strands are just in opposite direction. They aren't parallel. Okay, they aren't parallel. That is the meaning. Okay, so this strand which runs from 3' to 5', this strand is known as the antisense. Okay, this strand is known as the antisense, or the non-coding strand. Non-coding strand, or the other name we can call it, it is simply known as the template strand. The template strand. So the antisense, the non-coding strand, or the template strand is just the strand which runs from 3' to 5'. Now let's look at this strand which runs from 5' to 3'. So this strand is basically known as the sense strands. Okay, it's just the opposite. Here we've said that the antisense strand, is the strand which runs from 3' to 5'. From 3' to 5', that is the antisense. So the opposite is true for the strand which runs from 5' to 3'. So that strand is known as the sense strand. Sense strand. Sense, which means it has sense. Why is it called the sense strand? This because it contains the information. It contains the information that has to be coded. Okay? No wonder it is also known as the code strand. It has sense. It has information which has to be what? Coded. Okay? So basically that is a simple understanding you can get about these two strands. Okay, then the other way, or the opposite of temperate strand, when something is temperate, then the opposite of this temperate strand will be what? Will simply be the non-temperate strand. Okay, so the strand which runs from 5' to 3' will be called the non-temperate strand. Okay, so this will be called the non. So 3' to 5', we said it's a temperate strand, so this will be a non-temperate strand. Template strand. Okay, so this is simple. This is easy to understand. It is very easy, right? Have you seen how easy it is? Okay, it is very easy to understand. Okay, now let's look at something interesting here. Let's look at something very interesting. So I'll give an example. Okay, examples. Let's look at an example. Let's say we have a temperate strand. So we have a temperate strand here. I'll just write it in short. Okay, so this temperate strand we have, it is 3', then it has adenine, it has adenine, then it has thymine. Well, let's start with thymine. Okay, let's say maybe it has thymine, it has adenine, it has which nitrogenous base, it has cytosine, it has maybe, let's say, another cytosine, it has maybe guanine, another guanine, then it has adenine, Then let's say it has thymine, and let's say that is the 5' end. Okay, then how do you draw? This is the temperate strand. How do you get the non-temperate strand? How do you get the non-temperate strand? So something interesting here happens. Okay, something very interesting. Something interesting happens here. Okay, let me use a different thing. So here, the 3' end, the complementary end for 3' is 5'. Then the complementary for for thymine is simply adenine. For adenine it is thymine. For cytosine it is guanine. For cytosine again guanine. For guanine it will be cytosine. Okay, to be cytosine. Then for guanine again cytosine. Then for adenine that is thymine. For this is for thymine, that is adenine. For thymine, adenine. The 5' the complementary is 3'. So this is for non-temperate strand. Now let's look at for mRNA strand. Okay. Let's look at for mRNA strand. mRNA strand. Okay. For mRNA strand. So how do you get for mRNA strand? You need to understand that for mRNA strand, that is just for non-temperate strand, but we need to extract something there. From the non-temperate strand, we need to remove something. So it will simply be 5'. Adenine is present in mRNA, but thymine is not found in RNA. So what is found in RNA is uracil. So to be replaced by uracil. Guanine, cytosine. Then what else do we have? Thymine, it is not there, so to be replaced by uracil. Then we have adenine, we have thymine, is not there, to be replaced by uracil. Okay, so to be replaced by uracil. Then what do we have there? In fact, that was supposed to be, since this is thymine. Okay, it was supposed to be adenine here. Then 3'. Okay, so we're going to have uracil there. Then we'll have the 3' there. Okay, so this is basically how you can write the mRNA strand. It's very easy, right? It's very easy. Now let's look at the third process, which we said termination. Okay, the third process, which is known as termination. So for termination is simply the what? The ending. Termination is simply the ending. Okay, but before we can even end, we know that during translation there's what is known as what are known as sigma bonds. Okay, the what are known as sigma bond. So what this sigma does, it's just that this sigma is able to direct. Okay, it is able to direct the polymerase to the right side. Okay, right side of transcription, or it is able to direct it to the correct what area. It is just able to direct to the correct area of replication. Okay, so this one just directs to the correct area of what replication. Is able to guide that enzyme which carries out transcription to the right direction, to the right location for transcription to occur. Okay, now let's look at termination. Okay, we've said termination is just the ending. Okay, we've said the termination is simply the ending. You are now ending the chain. So what should you understand about termination? So termination, you need to understand that since termination is ending, there are two ways in which termination can occur. Okay, there are two ways in which termination can occur. So the first way is by termination sequence. Termination frequency. Now, what is it about a termination frequency? So about a termination frequency, there has to be a termination signal which has to encode. Termination signal encodes. This termination signal is like a hairpin. I know you guys are confused. For boys, they are confused. Okay, should I say men? Men are confused because they haven't seen before an hairpin. But this can be best understood by girls who uses these things. Okay, who uses my hairpin, what and what. Okay, so this can be understood by girls or ladies in short. You guys can be confused, but don't worry, you understand it at the later time, okay? But just know that it's like a hairpin. If you want to know more about it, ask a certain girl, they will explain what a hairpin is. Yeah, so it will be very difficult for you to understand termination. I know termination sequence, it will be very difficult for you to understand termination sequence, but just know that there's a signal. We call the termination signal which has to encode, okay? Which has to encode. This word should be encoded. There's a termination signal which has to encode, and that termination signal which encodes is like in form of a hairpin. Okay, but don't worry about the hairpin. You understand it later. Okay, let's look at another way in which termination can occur. Okay, so there is another way by use of a raw special protein. Okay, that is another way, a raw special protein. For a raw special protein, this is the one which just ends the chain. Okay, a raw special protein, it ends the chain. It is a certain protein. Okay, we know that enzymes are made up of proteins, right? Okay, let me not go into details about these things, but you need to know basically that there are two processes, or there are two ways in which termination can occur. One is by use of a termination sequence, and the other one is by a raw special protein. And for termination sequence, you need to understand that there's a certain termination signal which has to encode, and that termination signal is like in form of a hairpin. So for a raw special protein, this one is a protein, just more like an enzyme. Okay, more like an enzyme. So don't worry about this, but just know that the raw special protein is able to end a chain. Okay, is able to end a chain. We'll discuss more about enzymes. Now let's look at post-transcriptional modification. Post-transcriptional modification, right? Excuse me. We are looking now at post-transcriptional modification. So when you're talking about modification includes correction of errors. Okay, correction of errors, making something now more better, or okay, making something better in short. Okay, or more nice. Okay, making something nicer than it was. Okay, that is modification. You're now trying to make something you're now trying to modify something, to change here and there, to change some things here and there so that it looks nice. Okay, that is simply modification. When you're talking about post something which has now at the end. Okay, post at the end, like I'm sure you've heard of postnatal. There's prenatal, there's antenatal, then there's postnatal. So at the end, okay, at the end of something, post means at the end. So what happens during post transcription or modification? So the first thing that happens there is one, splicing of exons. Splicing, okay. The first thing is splicing of exons. What is splicing of exons? So when you're talking about splicing of exons, this is simply the cutting of exons. Cutting off exons, okay? Just simply cutting off the exons. You see, when you have this 5 prime, then let's say this is the strand, then we have 3 prime there. Okay, there are some things that has to be cut off. Let's say these are introns. Okay, these are introns, then these are exons. So introns do not code. Okay, introns do not code for a protein, so these are cut off. Then what remains are exons, so these are cut off. They are cut off. In everywhere you say introns, they are cut off. Okay, they are cut off. Then what remains are exons. Now these exons are the ones which code for information. So during post transcription or modification, the introns are cut off. Then the exons are the ones which code for information and are the ones which are joined. Okay, which are joined. So that is what you need to understand about that. The next thing you need to understand here is that there is this, okay, this substance known as the guanosine. Okay, there's this substance known as guanosine triphosphate. There's this substance known as guanosine triphosphate cap. Okay, what does this guanosine triphosphate cap do? You see, this is a strand running from 5 prime to 3 prime. So during post transcription or modification, guanosine triphosphate is added at the 3 prime. Okay, this is guanosine triphosphate. It is added. Okay, guanosine is just added at the 3 prime. Then on the 7th position, what happens during, or what happens at the 7th position? On the 7th position now, there is a methyl group which is added. A methyl group is added. So guanosine is added at the 3 prime. Okay, guanosine is added at the 3 prime. Guanosine triphosphate is added at the 3 prime. Now at the 7th position, there is another methyl group which is added at the 7th position. So what does it form now? When this methyl group is added at the 7th prime, okay, at the 7th position, it will form what is known as 7-methyl guanosine triphosphate. So you need to understand that. What have I said? I've just said that guanosine is added at the 5' end. Then at the 7th position, this methyl group is added. A methyl group is added, and this forms 7-methylguanosine triphosphate. As simple as that, right? So if they ask you at which position is the methyl group added, it is just at the 7th position. Then let's look at the last process which occurs. So the last process is known as polyadenylation. Okay, polyadenylation. Polyadenylation. There's adenine in that word. Adenylation. Polyadenylation. So you're simply talking about the adding of what? The adenine. So many adenine. Poly means many. So many adenine. Many adenine. Many adenine are added at what? At 3' end. Okay, so many adenine are added at the 3' end. There can be many, maybe even more than 250, or should I say about 250 adenines. Okay, just to give you an overview of how many are added. So maybe about 350. Okay, they are added at the 3' end, which is also known as the tail. So this is the tail at the 3' end. So when you have this strand, let's say that is 5', that is 3'. So they're added at 3' end. So we can have maybe adenine there, another adenine. There are many. Okay, there are many adenines which are added. There are a lot. And that is known as polyadenylation, because many adenine are added, or poly A. Even if you say maybe poly A tail. Okay, they're talking about polyadenylation, whereby many adenines are added at the 3' end. So let me test you with one question here. Let's say this question was asked in an exam to say mature mRNA do not contain what? Okay, mature mRNA do not contain what? Okay, or which one is not found in mature mRNA? What could be your answer? Okay, I give you 1 second to think about the answer. 1 second, or maybe 30 seconds to think about the answer. Mature mRNA do not contain what? Or what is not contained in mature mRNA? Okay, maybe you are still thinking, but whatever you are thinking about, okay, or whatever you think about, just know that mature mRNA do not contain introns. Okay, so that's an exam question actually, which was asked in one of the papers. Just try to check. I've just forgotten. My mind has misplaced the year, but it was one of the questions in an exam. Mature mRNA do not contain. Then there was dash, there was exons, introns, whatever. Okay, but the answer there is introns. Remember, we said that during splicing of exons, introns are cut off. These guys are cut off, so they are not contained in mature. They are just contained during premature stage or during the primary stage. Okay, so that's the end of RNA transcription. So that is the end of this topic. We have exhausted what needs to be exhausted. Okay, the next part we'll just do the exam question, more of the exam questions. So thank you for watching, and have a great day.